Welcome to video 3.7. In this video, I'll be covering tertiary structure of proteins, talking about two of the effects that matter, polarity and charge. Now, tertiary structure can be depicted fairly well, in my opinion, with this meme here. At this point, where we've drawn a couple circles, we have secondary structure, as we've talked about, the helices and beta sheets of a protein. And then tertiary structure is really just everything else happening all at once. Draw the rest of the effing owl. So tertiary structure is really the final structure of a single chain polypeptide. It involves the folding of the entire polypeptide, and it is much more complicated. Right? Modeling this is something that cutting-edge computer software is trying to do. However, it's still based on fundamentals. The idea that polarity, right, like dissolves like, charges need to match up, hydrophobicity, all of these things we've been talking about, these factor into the correct three-dimensional charge, or rather three-dimensional shape of the polypeptide. And for many proteins, that tertiary structure is its final functional form. So again, primary structure is the chain N to C of amino acids. Then it folds in simple and predictable ways to make secondary structure, so that's your alpha helices and maybe your beta sheets. Right here, only alpha helices are shown. It's H bonding of specifically the backbone that gives you the secondary structure, so that's all pretty predictable. And then here comes tertiary structure, where the whole thing folds together, and it's affected by all sorts of things, right? We have H bonding, we have charge, we have hydrophobicity, and a few other different factors that we'll talk about later that give you that final three-dimensional shape that then has its function. So with that in mind, let's talk about some key factors for tertiary structure in more detail, specifically related to the R groups. The first is the presence of full charges. So this is relevant when we're talking about our ionizable functional groups. Right? When we're talking about protein, remember the 8 or so charged amino acids that can potentially have charge. Amino acids like lysine and arginine carrying always a positive charge at pH 7. Amino acids like glutamate and aspartate carrying a negative charge at pH 7. Right? These charges can both attract. So, for example, arginine and glutamate would be attracted to each other, while lysine and arginine would be repelled from each other. Right? Positives and negatives attract, and two positives or two negatives will repel. Right? So here we sometimes see these linkages called salt bridges. Right? Because a salt has like a positive and negative ion, like NaCl. But we can see here that if we have two positive charges on this side of the polypeptide, they will not be close together. They're going to repel from each other. But if we see a positive and negative amino acid, or R group, right, near each other, they're going to be attracted, and that will help guide the three-dimensional structure. A second factor is polarity, right? R groups with H bond acceptors or donors. So for example, if we think about a few of these, serine with its OH, I think there's actually an extra carbon there, serine, threonine, right, tyrosine. In order to be stable, these OHs will need hydrogen bonding. And there's really two ways to get hydrogen bonded, right? Let's go ahead and erase this. The first is R groups. So other R groups in the same polypeptide chain. For example, here we see the delta negative oxygen, right? And the delta positive hydrogen making a nice hydrogen bond from different parts of the three-dimensional structure, right? They don't have to be close together. They're very far apart in the primary sequence, but they can be close to each other in space. They can make that H bond, and that can be something that keeps these molecules attached or more close to each other, and again, inform that three-dimensional shape. The other way you can think about H bonding is if you're on the surface of what's called a globular protein, which we'll talk about in a bit. The basic idea is that inside this glob of protein, it's very nonpolar, so you're not going to want to see OHs. Where on the outside, right, those OHs, they'd be stable because they're able to interact with water for their hydrogen bonding. If you put an OH in the middle here, there's no water in there, and so it would be very difficult. It'd be unstable for that OH to be there. We typically see things like alanine, right? Non-polar amino acids would be on the inside. So all of that together leads to something we'd call the hydrophobic effect, right? Non-polar groups prefer to hide from water. So we're thinking groups like alanine, valine, isoleucine, etc. Right, we talked about those in the previous video. I think it was video 2.3, where polar groups are going to want to actually be exposed to water. And this leads to what's, again, called a globular protein shape. So again, we can imagine that amino acids like alanine, leucine, phenylalanine, and valine are going to be found inside this glob, right, because they want to hide from water, where amino acids like serine, asparagine, histidine, and threonine here are exposed to water, right? This globular shape is what most proteins look like, although, of course, there are exceptions. And it's important to understand what factors, right, the hydrophobicity, the polarity of these proteins, the primary structure, forms this type of shape. Now, going even further down, some polypeptides actually fold into distinct structures. So we call these domains of proteins. And here I've kind of shown an example of this. So we have the red part, so this is the primary sequence, and we're going right to tertiary. So we're kind of skipping secondary sequence. But you can see here that the red part kind of folds into its own little thing here. And then there's some sort of linker, some sort of, black area, and then the purple side forms its own little domain. And usually each of these domains would have their own unique function. For example, maybe the red thing binds a red thing, and the purple thing binds a purple thing, and so those two things can be brought close together, right? Whatever the reason is, the idea is that when you see these folds, sometimes it's still a single protein. It came from a single polypeptide chain, but that doesn't mean that it can't have multiple functions if there are multiple protein domains. So again, usually each domain has a unique function. And the last thing to talk about before moving on to other types of factors affecting tertiary structure is this idea of motifs. So there are some kind of three-dimensional structures. So for example, we've talked about alpha helixes and beta sheets. These things are types of secondary structure that we see all the time. But there are some larger structures that we also see all the time. For example, here's something called the zinc finger. There's two alpha helices in a certain way. I'm not going to go into too much detail here, that specifically bind to zinc. Now, this zinc finger is actually really good at binding DNA. And you can imagine that there might be multiple proteins that want to bind DNA for different reasons. So it turns out if a protein is, if its function involves binding DNA to do something, it tends to have a zinc finger somewhere in its structure. If we think back to these multi-domain proteins, perhaps this could be a zinc finger that binds DNA, and this could be some sort of other subunit that does something else, right? So overall, it's just an idea here that we have these things called motifs, right? And domains exist, and there can be multiple domains with a single protein. Motifs are just common things we see across various proteins. With that in mind, hopefully you have a better understanding of the complexity that goes into tertiary structure. So we'll make it even more complex in the next video by discussing disulfide bonds and prosthetic groups. I'll see you there.